Okay, let's talk about Chapter 16. Chapter 16 is sort of bringing together 14 and 15 and talking about specifically how we regulate transcription and translation. So a lot of this should sound like a review because we did talk about it when we talked about transcription and when we talked about translation. Okay? So the overall message here is like we cannot be transcribing, translating, and expressing all these proteins all of the time. It would take way too much energy and there'd just be proteins everywhere. It would be a big fat mess. So we can't do that. There has to be a regulation. There has to be a way of turning the genes on and off only when we need the protein. So prokaryotic gene regulation is primarily at transcription, right? Because if it doesn't make a mRNA, then that doesn't get translated and proteins don't get made. Eukaryotic gene regulation is more epigenetic, the winding of the DNA, the histones, transcriptional, just like eukaryotic, post-transcriptional, how we splice our introns and extrons, translational, whether we do or do not make our mRNA into a string of amino acids, and post-translational in how we're going to fold our proteins. So eukaryotic gene regulation is a lot more complicated. There's going to be more chapters related to eukaryotic gene regulation. Okay, so again, each cell, somatic means not sex cells. So all of your other cells contain the full genome of an organism. All genes do not need to be expressed simultaneously. That would not be great. This is again for energy, right? It would just take way too much energy to express everything all at once. Where would we keep all of the proteins? And this way genes can be expressed more rapidly as needed. Okay, so here's just, right, a visual, the genetic content of somatic cells, aka every other cell that's not made by meiosis. So everything that's not your sperm and egg cells. But all genes are not expressed all the time because it would be exhausting. Right? We can't do that. Okay. How does the regulation benefit the organism? Again, prokaryotes, transcription and translation occur simultaneously. So when we don't need the protein, they just send a message that's like, hey, turn that off. We don't need that anymore. We're good. Thank you. The primary method of control, therefore, in prokaryotes is transcription. Because remember, it doesn't have to go through anything from transcription to translation. There's no moving it out of the nucleus, cap and tail. We're not doing any of that in prokaryotes. So the primary method then would just be to control transcription, because if there's no mRNA, then there's no translation. So everything happens. So eukaryotes have a lot of things going on. Prokaryotes, the answer is easy. How do we regulate gene regulation in prokaryotes? Transcription. That is the answer. Right? Because transcription occurs simultaneously. Therefore, it's best to just regulate it at the transcription level. The general model of transcription and translation in eukaryotic organisms, again, right, all happening all at the same time here in a bacteria cell versus a eukaryotic cell. We transcribe, and then we process that, cap it, tail it, take the introns out, get it all ready, process it out of the nucleus, and then translation happens. Here's a happy little chart, a little table, if you will, to look at the differences between prokaryote and eukaryote organisms. Okay, so let's talk a bit about prokaryotic gene regulation. So things that you need to know, this will be on the study guide. Steps involved in prokaryotic gene regulation. The roles of activators, inducers, repressors. And you will define operons and distinguish between repressor operons, positive regulators, and inducer operons. Okay? It's not as bad as it sounds. Prokaryotic DNA is organized in that circular chromosome. Remember, it's kind of a big circle, and you could have something transcribing right here. Proteins with similar functions or in the same biochemical pathway are organized in blocks called operons. Operons are regulated 3 ways. repressors, which suppress transcription, activators, which increase transcription. You should probably get those two on a multiple choice exam, right? You should get that without me even telling you. Inducers, that's where it gets tricky. They may suppress or activate depending on the needs of the cell. So they are there to figure out what do we need, what do we not need, when do we need it. Okay, starting out strong, the trip operon. This is a repressor operon, okay? When tryptophan is plentiful, there's a lot of tryptophan, okay? So tryptophan comes up here and binds this repressor. When it binds the repressor, we're not making any more tryptophan because we don't need any. Because we have enough free-floating tryptophan that this guy binds to this repressor and right at the operator, and that's going to prevent RNA polymerase and transcription, okay? This is a complex here that physically blocks RNA polymerase from transcribing the tryptophan.
Is a bacterial system for regulating the synthesis of the amino acid tryptophan. The operon controls expression of 5 genes. These structural genes are transcribed when RNA polymerase can bind the promoter controlling them. But when the cell has lots of tryptophan, why waste energy making more? The trip operon is at one position on the circular bacterial chromosome, and a regulatory gene for the system is about a quarter of the way around the chromosome from it. This gene, called TRPR, encodes a dimeric repressor protein on a slow but ongoing basis, and the repressors diffuse through the cytoplasm. We'll see in a little bit how the repressors work. Now let's look at the control sequences for the TRP operon. They include a promoter, the binding site for RNA polymerase, an operator, which overlaps the promoter, and a leader gene, TRPL, which has an attenuator sequence, which we'll also look at soon. When the bacterial cell's tryptophan levels are low, RNA polymerase can bind the promoter and transcribe the structural genes. Each gene encodes a protein chain for an enzyme in the TRP synthesis pathway. TRPE and D for anthranilate synthase, TRPC for indole glycerol phosphate synthase, and TRPB and A for tryptophan synthase. As tryptophan is synthesized, it diffuses through the cytoplasm. When it encounters a TRP repressor, it binds a site on the repressor, changing that protein shape. The repressor can now bind the operator, block polymerase binding, and prevent transcription of the structural genes. Thus, tryptophan is said to be a co-repressor. Like the LAC repressor, the TRP repressor does not stick like glue, but periodically dissociates, allowing a low basal rate of transcription. The operon can fine-tune tryptophan synthesis. In essence, it can read various levels of tryptophan concentration in the cell. It uses a system called attenuation to do this. When the tryptophan level is low, ribosomes translating the leader mRNA as it emerges from the RNA polymerase pause briefly when they encounter 2 tryptophan codons in a region called 1. They wait for TRP-charged tRNAs in order to continue. And while they wait, a pair of complementary regions of the mRNA, called 2 and 3, form a stem and loop. The next region that could have paired with 3 is not yet transcribed. When the TRP tRNAs appear, translation continues through the structural genes, and thus more tryptophan is produced. If, however, tryptophan-charged tRNAs are plentiful, there is lots of tryptophan in the cell, the ribosomes read through region 1 quickly, which allows region 3 and newly transcribed 4 to pair. This stem and loop terminates transcription, and the structural genes are not transcribed. Tryptophan is not produced, and its concentration in the cell decreases.
Okay, so this is a simplified version from the video. When there is a lot of tryptophan, it's going to bind to this repressor, changing it, and that is going to prevent, let's come here, that will prevent RNA polymerase from binding to the operator and it will prevent transcription. The absence of tryptophan, like we don't have enough tryptophan, right? This tryptophan that was here needs to be used. We don't have enough. When tryptophan is absent, the repressor protein does not bind to the operator, and RNA polymerase can access the operator, right? It can bind right here and it can start the process of transcription. Okay, so that is called a negative regulator. Negative regulator. CAP. So when glucose levels fall, E. coli may use other sugars for fuel, but must transcribe a new gene to do that. Okay, so there's not enough glucose, and E. coli's like, it's fine, I can use something else, right? I'll use some other, something else in the absence of CAMP, in the absence of glucose. Okay, so there's not enough glucose. Glucose supplies become limited. Therefore, CAMP levels increase. Okay, CAMP binds to the CAP protein. So this is our CAP protein. It's going to bind to the promoter, and what's going to happen? Binds to an operator region upstream and is required to use it. It's a positive, right? When it binds, it makes the transcription. Okay, therefore it's a positive regulator. Let's look at one last one. This is the lac operon. Okay, this is an inducer operon. In the absence of lactose, okay, so we don't have lactose, the lac repressor binds the operator and transcription is blocked. So in this case, if you don't have lactose here, the repressor is like, we're not drinking lactose. We don't have lactose. We don't need to make lactase. We don't need to make an enzyme that breaks down lactose if there's no lactose. Does that make sense? I mean, it kind of makes sense. It makes sense. Don't drink milk for a month and then go drink a bunch of milk and you will experience this. Even though you are a eukaryote, you will experience your body stopped making lactase because why? Same thing here. Why? You don't need it. So the repressor just sits here unless you have the presence of lactose. Okay? Inducible. It is induced by lactose. Okay? Activation and or repression depends on the local environment and the needs of the cell. So here again is the lac operon, and I think I have a video. Hold on.
Or basal levels. The lac operon is a bacterial system for regulating genes that control the utilization of a disaccharide, lactose. The operon controls expression of genes for beta-galactosidase, which splits the disaccharide, permease, which transports lactose into the cell, and transacetylase. These structural genes are transcribed when RNA polymerase can bind the promoter controlling them. When there is no lactose in the cell's environment, it would be wasteful of the cell to make these proteins. The cell keeps the RNA polymerase binding site turned off. Let's see how. Upstream from the lac operon is a regulatory gene, LAC-I, which encodes a repressor protein. The repressor tetramer is slowly but continuously produced by the cell. Now let's look at the control sequences, the promoter and operator, for the structural genes. The operator overlaps the promoter. If a repressor molecule is present, it can bind the lac operator. It doesn't stick to the DNA like glue, but continually binds and releases, so the operator is not bound by the repressor at all times, and structural genes are transcribed at low or basal levels. When there is little lactose present, there is no reason to waste energy making the enzymes to process it. But what happens when there is a lot of lactose to process and the cell must use it as its carbon source? The repressor proteins have sites that bind lactose. Lactose binding causes a change in the shape of the repressor so that it can no longer bind the operator. Without competition for the promoter, RNA polymerase can repeatedly transcribe the structural genes, and translation of the now abundant mRNAs makes the enzyme for processing lactose. The lac operon is an inducible system. and lactose is its inducer.
Okay, so that was the video here. Again, promoter, operator, repressor. Okay, and next slide. So this is just a question. I'll tell you the answer, but feel free to pause it and try and think about it. In E. coli, the tryptophan operon is on by default. So it is on kind of all the time. And then when there's too much, it turns it off. Versus the lac operon, which is off all the time and is only turned on in the presence of lactose. Think about it. Think about it in your own body. If you were in it, think about your day. Okay, pause it, try and guess, and then come back. Okay, I assume you paused it and you're back, or you thought about it for 2 seconds. Tryptophan is an amino acid. You need it all the time. So maybe you don't need a lot of it, but you do need it. So you need a base level of it all the time. Versus lactose, if you're not drinking milk or there's not a presence of lactose, you don't need an enzyme to break down lactose at all. Like, you just don't need it. Versus you do need some low level of tryptophan. So you do need the amino acid tryptophan. So it kind of, its default is on. Versus you may never need, people are lactose intolerant. The bacteria are lactose intolerant, and it's fine. So in this case, the E. coli may never come into contact with lactose, so it doesn't need to be on ever. Okay, that's that answer. And this is the end of prokaryote regulation, and we will have another video on eukaryote regulation.